So now that we're familiar with all the different groups of plants, at least terrestrial land plants, the bryophytes, ferns, gymnosperms, and angiosperms, let's talk a little bit more about plant structure. Now, most of this information is really kind of using angiosperms as the example because that's our most familiar plant. And so it doesn't mean that this isn't applicable to the other groups, but most of the stuff is going to use angiosperms as the example. So every plant has a shoot system and a root system, and that shoot system is going to speak to how a plant grows, as does the root system. So plants grow differently, and we'll talk about that in a separate lecture, but once we understand the parts of it, then we're going to understand how they grow. But basically, the shoot system is made of the stems and the leaves. And the distance between where the leaves branch or where the stem branches are called internodes. And the points on the stem where it branches or leaves form is called a node. So this kind of node-internode pattern is what we see for plants as they grow. Now, there's things called meristems, which we'll talk about in just a second. See here. These are clusters of cells at the tips of the shoots and roots. And this is where cell division is occurring. This is truly where a plant's growing. So whereas in animal cells, which don't have a cell wall, plant cells have cell walls. And because of that, you have to lay down cells kind of like you lay down bricks to build a chimney, as opposed to animal cells where, or animal growth, where cells can move and laminate and delaminate and migrate, but not so much in the plant cells. So that's going to affect kind of how it grows and what it looks like. So we've got node, the internode, we have the leaf. The leaf is photosynthetic organ. It is oftentimes made of what's called the petiole and the blade being the flat portion, the petiole being the base which connects the blade to the stem. Now we also have what are called axillary buds, and we contrast that then with the meristem at the tip of the growing shoot. So the meristem at the tip of the growing shoot is called the terminal bud, or the shoot apical meristem, the SAM. And the axillary buds are kind of meristems in reserve that will begin to grow for branching later if the plant is programmed to do that, or if you lose the terminal bud, another axillary meristem will take over. And those are always found at the node, kind of in the branching point between the leaf and the stem. When we look at the roots, we have a root system, very simple. There are different kinds of root systems. We'll look at variations on a theme in just a second. You sometimes have a taproot, but the growing tip of every root is called the ram, the root apical meristem. So this is the one on the right here now. And that is, you can see this very distinct root cap, kind of the first part of it on the bottom, and then there's that line. That is technically the meristem, but the root cap then is there to protect the meristem, the growing meristem, as it moves and pushes through the soil. Now, the thing that's cool about plant anatomy is that there are only 3 plant organs. That's it. Stems, roots, and leaves. Everything of a plant is one of these 3 things or a variation of them. And so the variations actually, which makes it a little tricky, are quite numerous. So for example, modified leaves. These could be the tendrils of a pea plant, the spines of a cactus. You have leaves that are very succulent for storage. They serve different purposes based on their modifications. Or even a poinsettia, which the leaves we think of as the flower of the poinsettia. The botanical flower is actually much smaller, but those red leaves look like flowers or petals to us, but they're in fact not part of the flower. So lots and lots of modification with leaves, lots of modification with roots. So we have things called stolons, which are above ground horizontal stems. We have rhizomes, which are underground horizontal stems. We have tubers, which are underground stems for storage, or you have the bulb, which is technically a very, very short stem with thick, thick leaves for storage. So lots of modification. And we'll play with this idea a little bit more and help you to identify if it's a leaf or a stem or a root. Now the roots, again, are modified. You can have a fibrous root system or a taproot system. You have things called adventitious roots, which grow from the stem and help support the plant. You have storage taproot, like think of a carrot as the kind of quintessential storage taproot. So it can get tricky when you look at compound leaves. You may say, oh, well, here's a branch. It's a stem with many leaves on it. In fact, that may be just all one leaf that is broken down into many different leaflets, right? So you talk about compound leaves and doubly compound leaves or thrice compound leaves. And I won't make you learn how to identify each of these. Don't worry about that. But I do want you to know, be aware that there's lots and lots of variation out there. But everything on the plant is either a leaf, a root, or a stem. Now, to exemplify that, let's look at some of the plants that you may be familiar with, and they're all derived from a common ancestor. They have all been bred to emphasize different components. So you've modified the stems of wild mustard, and you've got kohlrabi. You modify the leaves, you get kale. You select for the flower buds and stem, you have broccoli. For lateral buds, Brussels sprouts. Terminal leaf bud, cabbage. Flower buds, cauliflower. So all these are related, very, very closely related to each other, and they've all been domesticated to a certain extent by artificially selecting for a specific trait. Right? But they're all derived from wild mustard. So Brassicaceae is the family. Now, if we look at plants, we can break down their tissue systems into three main tissue systems. So there are three organs, and each organ is made of three tissue systems, right? So the tissue systems are the epidermis, or i.e. the skin of the plant, the vascular tissue, which we've been talking about, we'll go into more detail now, and then the ground tissue, which we'll talk about least, and that's all the space in between the epidermis and the vascular tissue. So let's look at the epidermis first, and in this picture that would be all the white parts of the plant, and literally it's the layer at the surface of the plant. That is the epidermis. That's very important because that's going to be interacting with the environment. Now, it's the first several layers of the plant, some of which then are going to secrete what's called a cuticle. And the cuticle is a waxy, waterproof layer, which is very thin on some plants and very thick on others. But here you can see the epidermal cells on the left, and that red stuff to the outside of them, that's the cuticle. Water on a leaf here on the right, you can see how it beads up because that leaf is waterproof. And a leaf has to be waterproof, right? Because if it wasn't, it'd be dehydrating constantly. And plants are already dehydrating constantly, which we'll talk about here in just a second. But you need to waterproof this so that you can at least control the rate at which water leaves the plant. All right? So if you waterproof the plant, if you cover it in this waxy cuticle, that's great for keeping water in. But just like we talked about the selective permeability of a cell membrane, going way back to the beginning of the quarter here, you can't have an impregnable wall. You've got to be able to move things in and out. So just like a cell membrane has proteins to make it selectively permeable, plants have holes or gates in oftentimes the bottom side of the leaves, which are called stomata. So the stomata are going to open and close depending on whether the plant needs to have gases move in or out of the plant. And just like you, the plant needs to have gases moving in and out of it. Remember, photosynthesis. What does photosynthesis use as a reactant? CO2. It needs CO2. So you have to let CO2, gaseous CO2, into the leaf, and it produces oxygen as a waste product, so it needs to get oxygen out of the leaf. All right. So, but it also needs to be able to say, oh, I'm getting too low on water. I need to close down those stomata because I'm losing water as I lose oxygen, and that's no good, right? So the stomata then are the control points, like I said, oftentimes found on the underside of the leaf in the epidermis that allow the control of gas exchange. So guard cells are the two cells that flank that hole that is the stoma and control it. Now, the vascular tissue of the plant in this picture is purple in color. And this is going to be made up of two types of tissue. That's the xylem and the phloem. Now, the xylem is going to be responsible for moving water around the plant, and the phloem is going to be responsible for moving sugars or photosynthate, the products of photosynthesis around the plant. And that's going to be food for the plant. All right? So xylem is water, and phloem is food. Now, the xylem is very interesting tissue because, in fact, it's dead tissue. So the plant will grow these cells, and literally it grows them into these long tubes, and then it kills them off. And so what remains is the cell walls. And those cell walls work like very effective tubes, and they can be impregnated with what's called suberin to make them much more waterproof so they don't leak so much, and they can literally draw water up from the roots of the plant all the way to the leaves through these tubes. So tracheids and vessel elements. Gymnosperms have only tracheids, and vessel elements are only seen in angiosperms. But this is also then the source of what we think of as wood. So plants are going to layer lots and lots of vascular tissue to move water from the roots to the shoots, and that's going to thicken the stem. And that thickening is called secondary growth, and that's all the wood that we think of when we think of like cutting down a pine tree or an oak tree for wood. All right. So literally, these are going to form these transport vessels, like to move the water, and that's called transpiration. So transpiration is going to be moving water from the roots to the shoots and through the xylem. Now, the water then comes out of the xylem elements where it's needed, because water always goes to where it's not. So a lot of this process is going to be driven by physical laws that we talked about with diffusion. And we're going to come back to this slide when we talk about phloem in just a second. But if you think about trees, they're very, very tall. So you've got to move that water a very long way. How do you do that? They don't have a heart like we have to pump our blood around. In fact, what they use is osmosis to a great extent. They use water potential. The fact that there's less water in the air around the tree, there's less water in the leaves than there is in the ground around the roots. And so water wants to move where it's not. So water moves in through the roots into the xylem, up through the tree into the leaves. And as the interior of the leaves, the water is saturated there, as it evaporates at the air-water interface and moves out of the leaf, it literally pulls water molecules up as it does so. So the surface tension of the water in the leaves actually pulls water up from the roots. Why does that work? Because water is weird, right? So let's go way back to the beginning of the quarter and think about water. Water is cohesive. Right? Water is adhesive with hydrophilic substances. So it wants to bind to the xylem, it wants to bind to water, and so when I tug on the top of the water column, it pulls other water molecules along with it. So this water movement driven by evaporation in the leaves is called transpiration. Okay, very important. So that light energy that's coming down and literally heating the leaves and driving transpiration to a great extent is also then the energy source for making sugars. We talk about photosynthesis occurring. So photosynthesis is making all these sugars, and the whole plant needs to eat, right? Especially the roots. Like, they don't get any sunlight. How do they make their food? Well, we've got to move the food from the source of the food to where it's needed, the sink. We call it from source to sink, where we have it to where it's needed. And we use source to sink, like a sink, because it's going to flow from areas of high concentration to areas of low concentration. So the phloem, let's go back to this slide here, the phloem is going to be a living system of cells for transportation of food. Now, what we have is leaves in the source cells in the leaves, which are actively loading, taking energy to load up a high concentration of sugar into the phloem. Now, water wants to go where it's not. So as we build up sugar concentration there, osmosis is going to draw water into the phloem, and that increases the pressure in this tube. So this tube then, if you direct it, if you say, oh, I'm building all this pressure, there's only one way to go, it's down towards the sink, right, or up towards the sink, depending on where it is, if it's the growing tip of the plant, that pressure will create bulk flow. And you literally then force the sugar from the source to the sink, where it's actively unloaded, and as the concentration decreases, water is more abundant there, so it moves to where it's not, and so you have this kind of osmotically driven pump, in essence, that moves sugar from the source in the plant to the sink in the plant. All right, so it's very interesting because it's not muscle driven, but it's very, very effective of getting things around a plant. Now, our last type of tissue system in the plant is the ground tissue system, and that's everything we see here in yellow, and that's going to be filling in the gaps between the epidermis and the vascular tissue. So oftentimes, if we see here this cross section of a leaf, it's these columnar, what we call parenchyma cells. So we have palisade parenchyma and spongy parenchyma. You don't have to worry about the details there, but this is the tissue, this is the mass of tissue that's actually doing photosynthesis in the leaf. So this area in between is called the ground tissue. It can be used for storage, it can be used for photosynthesis. It's going to fill in the gaps, All right, so we're going to keep that in mind as we think about the structure of a plant. Now, if you look at a leaf in itself, it's actually designed very, very well to be driven by gradients, right? So if you look at that cross section of the leaf, you have the upper epidermis, and then the palisade parenchyma, the spongy parenchyma, and the lower epidermis. So the way the leaf is oriented, most of the light is coming from the top, where you have massive amounts of photosynthesis going on. So you're going to be drawing up CO2, which is going to create a deficit of CO2. Things move to where they're not. You open up the stomata, the guard cells open up, stomata forms, you get CO2 moving down its concentration gradient. Lots of photosynthesis in the top of the leaf, lots of oxygen being produced. It's more concentrated in oxygen there. It moves down its concentration gradient to exit the plant. Water, it's 100% humidity inside the leaf. It wants to move where it's not. It moves out of the leaf, drawing water molecules with it. Transpiration is occurring. So if you think about the engineering of a leaf, it's has evolved well, obviously, to create efficiency to do the biochemistry that it needs to do to survive.